Good morning, all of you. Fine. So, last session we talked about the entire DNA recombinant technology in detail. I hope you have read that well. Very important part of the chapter. You can definitely expect one question in the NEET coming from the technique of DNA recombinant technology. Now we have an important concept, preparing and cloning a DNA library. So what is a DNA library? A library is a collection of DNA fragments, especially genes, from a particular species that is stored and propagated in a population of microorganisms through the process of molecular cloning. तो किसी भी एक ऑर्गेनिज्म के फ्रैगमेंट्स ऑफ डीएनए, टू बी मोर स्पेसिफिक जीन को प्लाज्मिड्स में डाल के उसे बैक्टीरिया के अंदर मल्टीप्लाई किया जाता है, which is called as making a gene library or a DNA library. There are two main types of DNA libraries, something called as genomic library and something called as cDNA library. So you have genomic library and cDNA library, and सबसे पहले we have genomic library. It is a collection of all the clones of DNA fragments that represent the complete genome of an organism. तो एक organism से humans, we are having a cell के अंदर nucleus के अंदर we have two DNAs. Both DNAs are called as genomes. One DNA is called as haploid genome, the other DNA is called another haploid genome. So we take one DNA out of the cell, और एक DNA यानी एक haploid genome को निकाला जाता है. We use molecular scissors, restriction endonuclease enzyme. Say we take EcoR1 and we cut the DNA at specific restriction sites into fragments. So we using a restriction endonuclease enzyme और DNA के multiple fragments मिलने लगते हैं आपको. Each fragment is taken and now it is put into a suitable vector. Sir, याद है ये, अगर एक जीन होता था, we would have used a plasmid. But a fragment is a collection of many genes, so we will use another vector. I hope you remember BAC or YC, bacterial artificial chromosome or yeast artificial chromosome are used, and sir, इसके अंदर बड़ा fragment of DNA is put inside, say in a bacterial artificial chromosome, and then this chimeric DNA or recombinant DNA is put into E. coli bacteria, and the bacteria replicates along with it. The bacterial artificial chromosome replicates and the fragment of DNA replicates. तो सर, हमने फर्स्ट फ्रैगमेंट को एक BAC में डाल के एक E. coli में डाला, उसके मल्टीपल कॉपीज बनाए, and we label that as fragment number 1. Similarly, fragment number 2 को BAC में डाल के बैक्टीरिया में डाल के उसके multiple copies बनाओ and label it as fragment number 2. Similarly, fragment number 3, fragment number 4, and the entire human DNA के जितने भी fragments बनते हैं, सब fragments को एक-एक E. coli एक-एक BAC में डाल के E. coli में मल्टीप्लाई करके उसकी एक पूरी लाइब्रेरी बनाते हैं. That entire library is called as the genomic library. तो पूरे ह्यूमन जीनोम के फ्रैगमेंट करके एक-एक फ्रैगमेंट का मल्टीपल कॉपीज बनाया जाता है, which is called as a genomic library. But sir, this library is not really of anything else. एक academic interest है कि हमने human DNA का library बना दिया. But mainly we are not interested in the entire DNA. If you remember, 98% of this DNA is junk. Only 2% are genes. And our interest is making use of genes or genes की library बनाओ. तो पूरे डीएनए का मतलब नहीं है, वी आर ओनली इंटरेस्टेड इन जीन लाइब्रेरी. So another concept which is called as cDNA library is an excellent library. So sir, what is cDNA? cDNA is complementary DNA, and complementary DNA is produced by using mRNA by a process called as reverse transcription. तो cDNA बनाने के लिए mRNA यूज करके रिवर्स ट्रांसक्रिप्शन यूज किया जाता है. This process is called as Teminism, named after its discoverers Temin and Baltimore. So what is cDNA library called as? Teminism, also called as the reverse transcription process made by Temin and Baltimore. Mostly for eukaryotic organisms, a cDNA library is constructed. So sir, mostly for us eukaryotes, which library is of more economical importance? cDNA library. Now what is the idea? Sir, in the DNA, a gene will undergo transcription to form an mRNA. तो एक सेल के अंदर डीएनए से बीटा सेल टेकन, उसके अंदर डीएनए में जीन फॉर इंसुलिन विल अंडरगो ट्रांसक्रिप्शन टू फॉर्म एन mRNA. This mRNA is removed out of the cell, and outside the cell mRNA is treated by an enzyme called as reverse transcriptase. What is reverse transcriptase going to do? Reverse of transcription. So what was transcription? DNA makes mRNA is called transcription. mRNA makes DNA is called reverse transcription. तो हमने जीन फॉर इंसुलिन का ट्रांसक्रिप्शन करके mRNA बनाया और उसको आउट ऑफ द सेल रिवर्स ट्रांसक्रिप्टेज एंजाइम की मदद से mRNA is allowed to undergo reverse of transcription called reverse transcription, and the DNA is made out of it. So mRNA से DNA बनाया जाता है, and that DNA is taken, which is the gene for insulin. It is put into a suitable vector. अभी एक जीन है तो प्लाज्मिड is better और प्लाज्मिड में डाल दिया जाता है. It is put into a E. coli bacteria, and the bacteria replicates. Along with it, your chimeric DNA will replicate, and the gene for insulin will replicate, and we get clones and clones, many multiple copies of the gene of insulin. So what is this library called as? A cDNA library, complementary DNA library. So we had two libraries. Agar entire DNA is broken into multiple fragments, and each fragment is taken, put into a vector and allowed to multiply, and we label फ्रैगमेंट नंबर 1, फ्रैगमेंट नंबर 2, फ्रैगमेंट नंबर 3, एंड पूरे डीएनए के जितने भी फ्रैगमेंट्स है उसकी लाइब्रेरी बनाते हैं. देन एंटायर डीएनए, द फुल जीनोम इज रेप्लिकेटेड, व्हिच इज कॉल्ड एज अ जीनोमिक लाइब्रेरी. But if we focus only on a specific gene and allow gene to undergo transcription to form an mRNA, and mRNA we allow it to undergo reverse transcription to form a DNA, then that specific gene is taken and put into a vector, and a library is made out of it. Clones are made out of it in bacteria. That library is called as a cDNA library. तो हमने cDNA library की मदद से specific genes की library बनाई है. So we have taken gene for insulin. Similarly, we take gene for clotting factor 8. Similarly, we take gene for interferons. Antiviral proteins are called interferons. आजकल कोरोना चल रहा है. You can give interferons, which are antiviral proteins, which will work against the coronavirus. उसके जीन से ट्रांसक्रिप्शन करके डीएनए बनाया जाता, आरएनए बनाया जाता है, एंड mRNA का रिवर्स ट्रांसक्रिप्शन करके डीएनए बनाया जाता है. एंड सर, दैट डीएनए इज पुट इनटू अ प्लाज्मिड and cloned in bacteria, and sir, whenever you require just take from that clone of say insulin gene or clotting factor 8 gene and put it into a bioreactor, and bioreactor में put in nutrition medium and put in this plasmid and bacteria with plasmid containing gene for insulin, and you can do mass manufacturing of insulin or mass manufacturing of clotting factor 8 or any gene which is now made by cDNA library, उसका mass manufacturing किया जाता है using a bioreactor. So we had two libraries, genomic library and a cDNA library, and now we have an excellent process, something called gene amplification, which is called polymerase chain reaction (PCR). So gene amplification called polymerase chain reaction (PCR). Gene amplification is obtaining multiple copies of a known DNA sequence that contains a gene, and sir, एक ही gene की billions of copies बनाई जाती है. Using a single gene, we make billions of copies of that gene. That technique is called as gene amplification, also called as polymerase chain reaction (PCR). And how is this PCR done? PCR is an in vitro technique of gene amplification. So sir, cell के अंदर नहीं, cell के बाहर is an in vitro technique of gene amplification. It is a scientific technique of molecular biology used to generate billions of copies of a particular DNA sequence in a very short time. So in a short time, we can make billions of copies of a particular DNA sequence. It was first developed in 1983 by a scientist called Carey Mullis. And Carey Mullis did gene amplification or polymerase chain reaction. In 1993, Mullis was awarded the Nobel Prize for this work. So what are the basic requirements for a PCR technique? PCR technique में basic requirements क्या है? Sir, this is a PCR box, और PCR box में यहाँ एक opening होता है जिसके अंदर we put in the gene that is required to be amplified. So we put in the gene to be amplified, and along with it, what else do we put? Sir, सबसे पहले gene or DNA segment which is 100 to 35,000 base pairs in length to be amplified. So how much is the maximum length of the DNA or the gene to be amplified? 35,000 base pair. So usually 100 base pairs से लेके 35,000 base pair का एक DNA segment is put into the PCR box. Then we put primers, forward and reverse. Sir, I hope you remember when we did DNA replication when we were doing genetics, that when DNA undergoes replication, it forms a replication fork, and 3' to 5' 5' to 3' on the both strands, RNA primase will make a molecule called RNA primer molecule. RNA primase will make a molecule called RNA primer molecule, and RNA primer molecule will attract the complementary nucleotides. If you remember, activated complementary nucleotide, if you remember, DATP, DGTP, DCTP, DTTP, if you remember, phosphorylase enzyme की मदद से AMP, adenosine monophosphate, or ATP, adenosine triphosphate, ATP is broken to form AMP, and phosphorylase transfers 2 phosphate to AMP, which will become ATP. Similarly, phosphorylase transfers 2 phosphates from ATP and acts on GMP to become GTP. So GMP will become GTP, TMP will become TTP, CMP will become CTP, AMP will become ATP. I hope you all remember this, all of you. Please read that part well. So sir, what do we have? Primers. RNA polymerase की मदद से क्या बनते हैं? Primers. तो सर यहाँ जीन एम्प्लीफिकेशन में हम लोग PCR बॉक्स के अंदर DNA डालते हैं and then we put primers. Primers are ready-made packets of RNA primer. तो RNA primer के ready-made packets होते हैं forward and reverse primers, which are synthetic oligonucleotides of 17 to 30 nucleotides. So synthetic oligonucleotides of 17 to 30 nucleotides, and they are complementary to the sequences present on the desired DNA segment. तो desired DNA segment है, ये है desired DNA segment, उसके ऊपर ये primers attach हो जाते हैं, which are artificial RNA primer molecules. And RNA primer molecules के forward primers होते हैं and reverse primers होते हैं. So there are forward primers and there are reverse primers. And these primer molecules are put inside, and then we put 4 types of deoxyribonucleotides. 4 types के nucleotides डालते हैं, adenine, guanine, cytosine and thymine, but in activated form. In activated form, so we put DATP, DCTP, DGTP and DTTP. If you remember, who activates them? Phosphorylase. Phosphorylase activates all the 4 types of nucleotides, and we use all 4 nucleotides, DATP, DCTP, DGTP, DTTP, collectively called as DNTP. तो ये चारों टाइप के deoxyribonucleotides, DATP, DGTP, DCTP, DTTP is put inside, and then we put a thermostable DNA polymerase enzyme. So we put in an enzyme which will bring nucleotides. So I hope you remember, जब strands separate होते हैं, so both of them will act as templates. And what is constructed over here? RNA primer molecule. And then what is delivered to RNA primer molecule? Activated nucleotide. Who will deliver the activated nucleotide? I hope you remember, DNA polymerase. तो DNA polymerase लेके आता है. अभी एक प्रॉब्लम है, in this PCR technique, the temperature of the box is raised to 90, 95, 100 degrees Celsius. And if you remember, you have done it in biochemistry, all enzymes undergo denaturation at 60 degrees Celsius. And what enzyme are we using here? DNA polymerase. And DNA polymerase will denature at 60 degrees Celsius. तो आपका gene amplification PCR होगा कैसे? Temperature तो we have to raise in the box to 90 degrees. So Carey Mullis had this challenge, how to use an enzyme which can withstand a temperature of 90 degrees Celsius. So he found a very stable, thermostable DNA polymerase which can withstand temperature of up to 94 degrees Celsius, and that was an enzyme called Taq polymerase, isolated from the bacterium Thermus aquaticus. Hey, you had done this. Thermus aquaticus, नाम से मालूम पड़ रहा है, lives in hot thermal springs. And this bacteria multiplies in hot thermal springs, or thermal springs में the temperature is as high as 100 degrees Celsius. And still this bacteria multiplies in hot thermal springs. So sir, he took this thermophilic bacteria called as Thermus aquaticus, और उसमें से एक enzyme isolate किया which was called as Taq polymerase. It is a thermostable DNA polymerase. It can withstand temperature of 94 degrees Celsius. तो सर ये 4 चीजें आप PCR बॉक्स में डालोगे. What have you put inside? DNA, gene जिसको amplify करना है. Then we put primers. Primers are RNA molecules which are forward primers and reverse primers. They are 17 to 30 nucleotides complementary to the DNA, complementary to the gene to be amplified. Then we put in activated ribonucleotides, deoxyribonucleotides. I'm sorry, activated deoxyribonucleotides. adenine, guanine, thymine, cytosine in the form of DATP, DGTP, DGTP, DTTP, and we use an enzyme called as Taq polymerase or DNA polymerase from Thermus aquaticus bacteria, which can survive a high temperature of 94 degrees Celsius. इसको बैक्टीरिया के अंदर डाल दिया जाता है. In the box we put that. And third, now we start with the PCR. The vast majority of PCR methods use thermal cycling. Thermal cycling alternate heating and cooling. So alternate heating and cooling is called thermal cycling. The three essential steps of PCR technique are heat denaturation. So what is the first step? Heat denaturation. Second is annealing, and third is polymerization. So three steps are there. First is heat denaturation, second is annealing, and third is extension, which is also called as polymerization. Now what do you mean by this, sir? तो PCR box के अंदर आपने सब कुछ डाल दिया. Calculations are made in the PCR box. It is an automatic process. So sir, what will happen? सबसे पहले the temperature in the PCR box is raised to 91 degrees Celsius. तो temperature 91 degrees Celsius तक raise किया जाता है, and at that high temperature of 91 degrees Celsius, the gene which we had put inside, उसके hydrogen bonds will break. And as hydrogen bonds break at 91 degrees Celsius, this is called as heat denaturation. And double-stranded DNA, by breaking of hydrogen bonds, will become single-stranded DNA. dsDNA will become ssDNA, ssDNA. What is that process of breaking hydrogen bonds called as? Heat denaturation. At what temperature we will do it? 91 degrees Celsius. समझो इस DNA fragment में we have more of GC. So 80s, if you remember, are doublets, but GC we remember are triplets. तो अगर GC ज़्यादा है, guanine cytosine ज़्यादा है, then triplets यानी हाइड्रोजन बॉन्ड को ब्रेक करने के लिए और भी हीट चाहिए. Then we raise the temperature from 91 to 93 or 94 degrees Celsius. If there are more GCs in the DNA, then we use 93 to 94 degrees temperature and break all the hydrogen bonds, which is called as heat denaturation is done. Now, from high temperature 91 degrees, the temperature is lower. If you remember, this is thermal cycling. So high temperature now to low temperature, and we reduce the temperature, which is called annealing. Annealing में the temperature is about 255 degrees Celsius, and heat denaturation was at what temperature? 91 degrees. Annealing is done at what temperature? 55 degrees. On heat denaturation, the double-stranded DNA has now become single-stranded DNA, and when the temperature is reduced from 91 to 55 degrees Celsius, annealing will happen, in which the primers will pair to the single-stranded DNA segment. ये जो primers आपने डाल के रखे थे, these primers will get attached to the DNA segment. तो यहाँ primers, ये forward primers will get attached here, and यहाँ reverse primers will get attached here. And that attachment of primers is called as annealing, which will take place at 55 degrees Celsius. Then we again raise the temperature to 72 degrees Celsius, which is now polymerization. And polymerization में Taq polymerase or DNA polymerase adds the DNTPs to the primer molecule. So activated DATP, DGTP, DGTP, DTTP, इनको लेके Taq polymerase will start attaching the new nucleotide in the forward direction as well as reverse direction, and that means a new DNA strand is made and one strand has now become two strands. एक strand के अभी दो strand हो गए, and what is adding of new nucleotides called as extension, which is also called as polymerization. So sir, what are the three steps we have done? At 91 degrees Celsius, we have broken hydrogen bonds, which is called as heat denaturation. Double-stranded DNA now becomes single-stranded DNA. Now the temperature is reduced to 55 degrees Celsius. On the DNA strand, you will have RNA primer molecules, forward and reverse primers will get attached, and that is called annealing. And when the temperature is raised again to 72 degrees Celsius, Taq polymerase or DNA polymerase will start supplying new nucleotides to the RNA primer molecule in the forward direction as well as reverse direction. And so from one double-stranded DNA, we have now got two double-stranded DNA, means one gene has amplified to become two genes. And what is this beautiful technique called as? This is called as PCR, polymerase chain reaction. How much is the time required for this process, sir? These three steps constitute one cycle. Each cycle takes place in 3 to 5 minutes. सिर्फ 3 से 5 मिनट के अंदर the entire DNA strand is undergoes replication. The cycle starts automatically again. So sir, what is the first step? Heat denaturation. What is the second step? Annealing. What is the third step? Polymerization. And one DNA has become two DNA. Again it will undergo into heat denaturation, again annealing, again polymerization, again heat denaturation, again annealing, again polymerization. So cycle repeat every 3 to 5 minutes, and ऐसी 28 to 30 cycles होती है. 28 to 30 cycles के बाद the PCR progresses, the DNA generates itself as a template and exponentially multiplies. Can you imagine 30 cycles? एक cycle में 1 will become 2 DNA, then 2 will become 4, 8, 16, 32, 64, 128, 256, and ऐसे 30 cycles करेंगे. Do the math. Then one fragment would have become billions of fragments, and the time required would be just 5 minutes into 30 cycles. So approximately 150 minutes, which is 2.5 to 3 hours के अंदर, we have replicated one DNA to form billions of copies of a gene fragment. तो समझो हमने gene पर insulin लिया था, एक जीन ऑफ इंसुलिन से हमारे पास बिलियंस ऑफ जीन्स फॉर इंसुलिन हो गए by a beautiful technique called as PCR, polymerase chain reaction. Nice logic, beautiful logic. इसको याद रखना, बहुत crucial part हमने ये किया है. Right. And then we have applications of biotechnology in agriculture, BT crops. So, you use chemical fertilizers and pesticides for growing plants, DDT, etc., or beta-hexane, or DDT, artificial urea. All these are artificial chemical fertilizers and pesticides which are used. But nowadays, scientists are going in कि don't use chemical pesticides because ये हमारे body में भी आ सकते हैं, and TTT or DSC etc. accumulates in the body, so they are toxic to the body. So don't use chemical fertilizers. Use biopesticides and biofertilizers, which is called organic farming. So use compost. Use products of fields which are made into pesticides and fertilizers. उसी से आप farming करो, which is called organic farming. So no artificial pesticides or herbicides or fertilizers are used. We use natural compost or other organic fertilizers, which is called organic farming. And nowadays we have an excellent logic, use genetically engineered crop plants which are called as transgenic plants or BT crops. तो सर आजकल ट्रांसजेनिक प्लांट या बीटी क्रॉप्स को ग्रो किया जाता है which are genetically engineered crop plants. तो उसको जेनेटिकली इंजीनियर करके ग्रो किया जाता है and such plants are called transgenic plants or BT crops. Transgenic plants carry many desirable traits. They are disease resistant. तो उनको infection, genetically engineered plants don't get infected by diseases. They are insect resistant. तो उनके ऊपर locusts या कोई insect का problem नहीं आता है. And they are herbicide resistant. उसके ऊपर आपने कोई herbicide डाला, weedicide डाला, the weedicide will not kill the plant. So it's insect resistant, disease resistant, chemical resistant, herbicide resistant. Plants with transgenic plants have better photosynthetic efficiency. They do better photosynthesis. Their nitrogen fixing ability is excellent. They do excellent nitrogen fixation. They have improved storage protein and higher vitamin content. उनमें ज़्यादा improved storage of proteins होता है. Vitamin content is also high. So these are excellent transgenic plants, and ये सब start हुआ by using the first transgenic plant which was by BT, एक soil bacterium called Bacillus thuringiensis. सर ये तो नीट में इतनी बार पूछा है ना, ये तो याद रखना ही रखना. What are BT plants? BT plants are treated with a bacteria called as Bacillus thuringiensis. It is a soil bacteria. BT, Bacillus thuringiensis, is a soil bacterium that produces a protein with insecticidal qualities. And traditionally, an insecticidal spray produced from this bacteria is used. तो सर से हमारे इंडिया में, खास करके हमारे महाराष्ट्र में कॉटन बहुत कल्टीवेट किया जाता है. महाराष्ट्र इज अ स्ट्रॉन्ग बेल्ट फॉर प्रोड्यूसिंग शुगर केन एंड कॉटन. तो लॉट ऑफ कॉटन फार्म्स आर देयर इन महाराष्ट्र. अभी जब कॉटन ग्रो होने लगता है तो जमीन में से एक वर्म आता है, एंड दैट वर्म goes all the way up to the stem of the cotton plant and then goes to the leaf of the cotton and starts eating away all the leaves, and that worm is called as bollworm. तो ये bollworms cotton के leaves को खा जाते हैं. What will happen if bollworm eats away all the leaves? Then sir, cotton plant के ऊपर सारे leaves अगर चले जाएंगे, photosynthesis is not possible and the cotton plant will die. So you have to protect the cotton plant from this bollworm. तो bollworm से cotton plant को protect करना है, and how do you protect the cotton plant from bollworm? तो ये बैक्टीरिया दिया जाता है called Bacillus thuringiensis. उसको अगार अगार पे कल्टीवेट करते हैं and we put it into bioreactor, और बायोरिएक्टर में upstream process, that is making of the protein in the bacteria, and then we collect the protein out of the bacteria. I hope you remember bioreactors उसको निकाल के we will isolate a protein from the bacteria, Bacillus thuringiensis, that is called downstream process. And that protein is a protein called as cryo protein. What is a protein called as? Cryo protein. And cryo protein is made with the help of a gene which is called as the cry gene. तो ये है बैक्टीरिया Bacillus thuringiensis BT, उसके अंदर है DNA और उसके अंदर एक जीन है called as the cry gene, and cry gene undergoes transcription and translation to form the protein called as cryoprotein. So what is that protein called as? Cryoprotein. And cryoprotein is collected from the Bacillus thuringiensis and put into two cylinders. और सिलेंडर्स के ऊपर ये क्रायोप्रोटीन डाल दिया जाता है, उसमें डिस्टिल्ड वाटर रहते हैं या प्लेन वाटर रहते हैं, and these cylinders with cryoprotein are taken by a farmer and he uses a nozzle and sprays all this cryoprotein on cotton plants. तो कॉटन प्लांट्स के ऊपर एक क्रायोप्रोटीन स्प्रे किया जाता है, तो जैसे ही लीव्स के ऊपर क्रायोप्रोटीन आ जाता है, बैक्टीरिया में से बनाया क्रायोप्रोटीन and we have sprayed it as an insecticidal spray on the leaves of the cotton plant. Now, as soon as the bollworm comes and goes up the stem and eats the leaf, तो जैसे ही लीफ खाएगा, लीफ के साथ साथ bollworm के बॉडी में क्या जाएगा? Cryoprotein. Now, cryoprotein in nature is an inactive protein. It is inactive. And it does not affect the cotton plant because the toxin is inactive. But once the bollworm eats it, then in the alkaline pH of the small intestine, bollworm के small intestine में alkaline pH होता है. In the alkaline pH of the small intestine, the inactive cryoprotein now becomes active cryoprotein. So inactive becomes active cryoprotein, and active cryoprotein makes holes, pores in the intestine. तो intestine में pores होने लगते हैं, and as intestine में pores हो जाते हैं, absorption of food will not take place. There will be severe nutritional deficiency in the bollworm, and the bollworm, because of holes in its intestine, because of cryoprotein, will fall off the leaf and die. तो लीफ के ऊपर bollworm survive ही नहीं कर सकता है, and we have made this plant become resistant to the bollworm. तो हमने क्या किया? Bacillus thuringiensis के क्राय जीन से बनाया क्रायोप्रोटीन. क्रायोप्रोटीन को सिलेंडर्स में डाला and we sprayed it on the cotton plant. लीव्स के ऊपर क्रायोप्रोटीन आ गया, bollworm ने खाया क्रायोप्रोटीन. Cryoprotein went in the intestine. In the alkaline pH of the intestine, inactive cryoprotein becomes active and makes holes in the intestine, and that will cause death of the bollworm, and the plant has become resistant to bollworm. What is this called as? A cryoprotein induced treatment, which is called as Bacillus thuringiensis, BT plant. तो हमने कॉटन को बीटी बनाया है, which is called BT cotton. Similarly, we have made BT corn, BT rice, BT tomato, BT potato, BT soyabean. They have all become resistant to certain types of insects. So they have become insect resistant. And as a matter of fact, you may remember we have got 3 genes. 3 genes है. Cry1 AC gene and Cry2 AB gene. कौन सा जीन है ये? Cry1 AC and दूसरा कौन सा है? Cry2 AB. उससे बनाते हैं क्रायोप्रोटीन which kills cotton bollworms, जो cotton plants को इन्फेक्ट करता है. And there is another gene called Cry1 AB gene. Cry1 AB gene which controls corn borer. तो मकाई के दाने मेज कॉर्न के ऊपर एक इंसेक्ट आता है कॉल्ड एज कॉर्न बोरर, एंड कॉर्न बोरर बोर्स थ्रू द कॉर्न ट्रीज एंड कॉर्न प्लांट्स, और उनके फ्रूट्स के अंदर ये बोर कर देता है, एंड उसके अगेंस्ट एक क्रायोप्रोटीन स्प्रे किया जाता है. एंड विच इज द जीन विच कोड्स फॉर दैट सर? Cry1 AB makes the cryoprotein which will kill corn borer. So if you are understanding, this is very gene specific. Cry1 AC and Cry2 AB control cotton bollworm, and Cry1 AB produces cryoprotein which kills the corn borer. So this is the way Bacillus thuringiensis is very effectively used as a wonderful insecticide. Nice. Sure. We'll take a small break over here. I hope you have understood very well. We'll just start with the next part, a small break. Okay. Take care.